Hello, everyone. My name is Andrew Kim, and welcome to the very first lesson of the whole series for AP Bio exam prep. So the first unit will be water and macromolecules. Now, this unit will be a very fundamental unit that will set a lot of important information for later units. So I do strongly advise you to watch this video and take away some important points. So first of all, let's talk about water. Now, what is water? Well, we all know that water is H2O. And now, how this looks like is like this. Now, I know that this is not AP chemistry, so I won't be explaining about what each of the, signs and everything means, but there are still some important things that we have to know. Now, first of all, as you can see, H2, right? We have two H's, meaning that we have two hydrogens, and we have one O, meaning that we have one oxygen. Now, what these means is not very important, but what you have to know is that when you have multiple water molecules, let's say we have two of them, then what's going to happen is between these electrons, these two dots, and H you have over here, there will be a bond formed. Now, it's not going to be a bond where it actually makes the two molecules stick to each other. That's not going to happen. However, there's going to be this sort of sticky, this force that makes these two molecules sort of pull each other. So this is what it's called hydrogen bonding. And hydrogen bonding is an example of intermolecular force. Now, I said intermolecular, right? It's force between two molecules. So when you have two or more hydrogen molecules, then each of them will pull each other because of the water structure, how they have this H and these dots over here. So what we really have to remember is that water is sticky. Now, I know that there are a lot of other sticky liquids out there, but water, although it might not be relatively as sticky, but still, it pulls each other. I mean, think about it. When I was young, I used to, when we have these tissue pieces in the bathroom, I would like soak it in the water and make it kind of nasty and throw on the window. And you know how it would just stick on the window, right? So water has this inherent trait that it pulls each other and is very easy, okay? Now, this will cause a lot of important traits that water has, which will be explained later. But first of all, I want you to remember one more important characteristic of water is polarity. So the second characteristic I want you guys to remember is polarity. Now, polarity is basically, now did too much chem stuff, so I'll just summarize. Okay, now there are some molecules that are non-polar and there are molecules that are polar. Okay, now polar molecules like polar molecules, meaning that polar molecules will easily be dissolved in polar, for example, water. And this majority of the substances in this world are polar, majority of the substances will easily be dissolved in water. That is why water is called a universal solvent. Okay, so example, salts, NaCl, right? It's polar and it's easily dissolvable by water, right? So that also leads to a lot of important characteristics of water, which will be okay. Now, let's actually talk about these two, hydrogen bonding and polarity are two basic characteristics that lead to all the practical characteristics that water has. Now, let me explain some of those practical characteristics. First is adhesion/cohesion. Now, adhesion and cohesion is the biggest example of a trait formed by water's hydrogen bonding. Now, as I said, because of hydrogen bonding, water is sticky, right? Because water is sticky, cohesion is a force between water molecules. So this is literally what I just explained. Water molecules will tend to pull each other. That is cohesion. Then what is adhesion? Adhesion is a force between water molecule and a non-water molecule. For example, let's say glass. As I explained before, when you throw that soaked tissue onto a glass or a window, it's going to stick there. Why? Because glass is polar, because window is polar, mirror is polar. So it will stick. Now, as I said, when I was explaining polarity of a water molecule, polar molecules like polar molecules, right? Since water is polar, other polar molecules will easily be pulled by, attracted by, and stick to water. So water can not only pull other water molecules, but it can also pull other polar molecules. And this leads to one important characteristic of water, which is capillary force. Now, you don't have to remember this term, but just know what it is. For example, when we have a stem, we all know, learned from elementary school that plants have these stems and they also have roots, and they would, soak up all the water molecules from the roots, and the water molecules soaked up through root will go up the stem, right? Now, how it goes up the stem is basically, if you have water here, let's say you have water like this, okay? Now, if you look the water molecules here, it's going to be pulled by the glass molecules because they're polar. So water molecules will go up like this. But once water molecules go up, now the water molecule up above will pull the water molecule down. I mean, pull the water molecules that are down. So these water molecules will also be pulled by the water molecules up. So water will go up a little bit more. Now, once again, if water is like this, then the water molecules at the edge will be pulled by the glass molecules above. So it will go up. So it's going to look like this. Now, once we have this sort of round shape formed, then the water molecules above will again pull the water molecules down. So the water molecules below will also be pulled up from like being pushed above. Okay, so this is how capillary force, or the process of water traveling through stem, works. Now, then some people might ask, then why doesn't water on just a cup go through the same process and move above? Well, right, the forces in stem and plants, so there will be other forces that actually make this between glass or stem and water molecule, because they're both polar. It is happening. Okay, so just remember, molecules pull other polar molecules, which makes water molecules pull other water molecules, and also water molecules pull other polar molecules. Okay, now that first characteristic. Okay, so what is surface tension? Wait, well, basically, if you think about water structures, these lengthy bugs that look kind of like Aryan with long legs, you know how they literally just walk on water, right? How is that possible? Well, it's because, as I said, water is sticky. Because water molecules pull each other, it's kind of like a net. So if humans, we're heavy, right? So we can't just literally walk on water. These small and thin and very delicate water striders can actually walk on the net, because water molecules form this net because of its polarity, or because of its hydrogen bonding, which makes each one of the molecules pull each other. Okay, remember, it's specific heat. Now, specific heat, the definition of specific heat is how much energy you need to increase the temperature of a liquid by 1 degree Celsius or something like that. But that is too complicated, right? So just remember this: if you have high specific heat, be changed. Whereas if the specific heat is low, then the temperature will easily change. Okay, so this is kind of like, how easily will you change? If specific heat is high, then you'll easily change. If it's low, then you're not going to change easily. Now, water has a very, 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 I'm so sorry, I explained that opposite, okay? So if you have high specific heat, then you're not very likely to change your temperature. If the specific heat is low, then you're very much likely to change temperature, okay? So high specific heat means you're very resistant to heat, okay? So see this as sort of your power in how much resistant you are towards heat, okay? So water, again, has very high specific heat, meaning that it is very, very not likely to change its temperature, okay? So, and why is this happening? Well, okay, if we go deeper into, then changing temperature means molecules move faster, and since water is sticky, since water molecules pull each other, it's harder to make each one of them move faster. okay? But that's more of a chemistry stuff. You don't really need to know about that. But just remember, water molecules are, again, sticky. They bond to each other really easily, so it's like a net. Now, compared to just fluid, just freely moving molecules, if molecules are pulling each other, then it would be harder to make each molecule move faster and make its temperature go higher easily, right? So that is why water has high specific heat, meaning that it's very hard to change its temperature. Now, why is it important in biology? Well, it's because human body is formed 70%, the 70% of human body is water. Now, what this means is if water temperature can easily change, then 70% of our bodies easily change, mean that our body temperature would easily change. We will be like snakes. No, that should not be happening, right? That is why if water has high specific heat, meaning that water's temperature change easily, our body's temperature would also not change easily, and our body's temperature will maintain very, very steady. Okay, it's kind of like Mediterranean region, right? If you go to Greece, if you go to those European countries, even in the winter, the temperature doesn't go down too low. Why? It's because it's surrounded by ocean, right? And ocean is all water, right? So in order to increase those regions, you have to increase the temperature of the ocean, but that's hard. Why? Because ocean is made out of water, and water has high specific heat, meaning that it is hard to increase the temperature. Okay? So next time you visit Greece or whatever countries there, you can, kind of like have the bragging right? I know this, you know it. Well, don't be cocky like me. Okay, the next thing you have to know is, okay, this is not super important, but this might come up, so let's just know, okay? Which is hexagonal bonding. Now, hexagonal bonding does not exist in water, but it exists in ice. Now, when H2O molecules bond to form ice, it makes this weird hexagonal structure. Now, why is this important? Instead of just literally bonding like this, if you're forming these weird hexagonal structure, then you will have a bigger vacant area in the middle, right? Meaning that the volume will increase. Now, logically thinking, solid should have smaller volume compared to liquid. Why? Because liquid is flowing, meaning that there are more vacant area between each molecules, whereas solids are rigid, right? They're just packed with molecules. So the volume should be smaller. But when it comes to water and ice, it's opposite. Ice has lower density. It's lighter. Why? Because you have this vacant area in the middle. Okay, so ice is less dense than water. And why is this important in biology? Well, it's not very noticeable, but one part we can see this is pond. When we have a pond, if water starts, when it's winter, we often see the top layer of water being frozen. Well, we never see because we're living in Singapore, but when we travel to different countries or if we watch movies, we have these ice floating on water, and beneath the ice, it's all water, right? This is possible because ice is less dense, so it floats. Now, if this was not the case, if the water started freezing from the bottom because ice is denser than water, then what's going to happen is this pond won't have this top layer of ice that sort of protects the water below from freezing. It's kind of like losing a layer of blanket, meaning that the entire pond will end up freezing with all the fish, all the, organisms in the pond freezing together like a gigantic ice cream with just fish in the middle, which is bad for the ecology because every winter all the fish will die, right? But thanks to this water's characteristic of being denser than ice, all the ponds can have this extra layer of blanket on the top made out of ice, and the pond can just, stay as a liquid with all the organisms being alive, okay? Now, that is pretty much it when it comes to water, okay? So you just have to remember one thing. Hydrogen bonding happens, which makes water sticky, and that one characteristic leads to all these characteristics: surface tension, adhesion, cohesion, specific heat, and everything. Okay, hexagonal bonding is a little bit different, but still, most of the important characteristics of water comes from its hydrogen bonding and its polarity, okay? So just remember that. Now let's move on to the second part of this unit, which is about different macromolecules. Now, what is a macromolecule? What does macro means? Macro means big. Macromolecules are big molecules that are important to your body. For example, carbs, for example, fat, for example, protein, for example, okay, whatever, I can't spell, nucleic acid, and for example, what else do we have? That's pretty much it, right? 4 of them. Now, I'll explain one by one, okay? It's really important that you remember the major characteristics of each macromolecule, okay? And when I say major characteristics, I'm talking about their monomer, their polymer, their structure, and how it is used in biology or human body, okay? So first of all, let's talk about carbs. Now, what is carbs monomer? Carbs monomer is very easy to memorize, monosaccharide, okay? It literally says mono, so it's not going to be hard to memorize, right? Monomers of a carb is monosaccharide. Now, what is a polymer of a carb? Still very simple, polysaccharide, okay? Now, we will actually look at each of them. One example of a monosaccharide is glucose. And one example of a polysaccharide is sucrose. Then how are polymers made? Well, before that, what is a polymer and what is a monomer, Andrew? You nerd, why are you taking as granted that we already all know what monomer and polymer means? Well, don't worry, I'll explain. The monomer is like a Lego block, a singular Lego block, just one piece. Polymer is when you connect all the monomers, then you get an actual shape of a Lego, right? That is a polymer, okay? So monomer is like a component. Polymer is the whole structure that you build out of monomers, okay? Now, so how are these bonds made? How can each Lego block bond to another Lego block to make this polymer? Well, in the case of carbs, this linkage between two monomer is called glycolytic linkage. And this glycolytic linkage is made through a process called dehydration synthesis. Okay, dehydration synthesis. But of course, when we build a Lego block, we also sometimes want to, crash it, right? We want to unbond it. That unbonding process is called hydrolysis. Now, hydrolysis, dehydration synthesis, was number 1 in our AP Bio mock, and I got it wrong because I forgot it. But I hope if it's in the actual test, we all get it right? So I'll explain how this works. Now, if we have one monomer, another monomer, again, they're connected through glycolytic linkage, right? And that process is dehydration synthesis. Now, as you can tell from the name, dehydration, D means opposite, hydration means adding water, right? So dehydration means not adding water, but taking water, and synthesis means combining, right? So if you just take it from the name, dehydration synthesis means when we have two monomers, then we're taking water from the two to make a bigger piece. So it will look like this. Okay, you have two monomers, you combine the two, but you're taking H2O out of it. Okay, now this might look kind of weird because it says dehydration synthesis, but as a product, you're literally getting H2O, you're getting water. How is this dehydration? Well, you have to think from this way. These two monomers had water, right? But we're taking that water out. That is why we're receiving water as a product of this reaction, right? So if you think from the monomers perspective, they're losing their water in order to synthesize and form this bond to be a polymer, right? So let's remember that, okay? Dehydration synthesis happens by two monomers being combined as one polymer, but as a result, it's getting one H2O pulled out, okay? Now, hydrolysis is the exact opposite. Hydro, water, lysis. What does that even mean? I don't know. Hydrolysis means you're adding water so that one big polymer can split into two monomers. So of course, if you write as a form of reaction, it will look something like this, right? Equation, it's going to look something like this, right? You have one big polymer, and by adding an H2O to the polymer splits into two monomers, okay? So it looks like this. Now one biggest example is when you have two glucose. Again, glucose is a monomer, and if these two glucoses go through a dehydration synthesis, what's going to happen is a sucrose molecule will be made, but as a result, an H2O will also be made. Now, again, H2O is being made, we are having more water, but it's still called dehydration because you have to think from glucose's perspective. From glucose's perspective, they're being robbed in H2O in order to combine, right? They're losing water, dehydration, okay? So that's how dehydration synthesis works in order for a glycolytic linkage to be made and in order for a polymer to be ultimately be made, right? So those are the two things we have to remember. And now let's look at some examples of carbohydrates. One is, again, glucose. As I said, it's a monomer. And its role in our body is it's used as an instantaneous energy source. So the moment you eat it, it's instantaneously providing you energy. That is why if you eat high carb food, if you eat donuts, your blood pressure increases steeply, right? Why? Because you're eating glucose, because you're eating monomers that are used as instantaneous energy. Energy instantaneously goes up, your blood pressure will instantaneously go up. Okay, that is why monomers are bad, and that is why processed carbs, right? All the sugars, all the bad flours, those are really bad for your body because they're all monomers and they are instantaneous energy source. They will make your blood pressure steeply increase. Whereas we have things like glycogen or glycogen, however you call, glycogens or glycogens are not monomers, and also they mean that they're polymers, right? And they store energy for a longer term. Okay, so it's not instantaneous energy source, but it's a form of storing energy so that you can use it later. So it's not going to steeply increase your blood pressure. Now that is why I often say, sorry, I'm cutting. I'm trying to eat healthy. I'm trying to eat clean. I don't want to eat flour, and I start eating pasta. Then my friends tell me, why are you eating pasta? Pasta is literally wheat. It's flour. Then I say, oh, actually, pasta is carbs, but also at the same time, it's not a monomer. It's a polymer, so it's used for long-term energy. It's not going to increase my blood pressure steeply and make me fat and ugly. So I eat pasta and I'm keeping my diet, right? So glycogen. The last one is cellulose. Now, cellulose is used for cell wall. So in the next unit, we will be learning about different types of cells, cell structures, and one of them will be plant cells. And when we learn cells, we're going to learn about their cell wall as well. So cellulose will appear in the next unit. So just remember. Now let's move on. My least favorite, macromolecule, which is, of course, lipid. Now, I forgot to mention one really important thing. Now, we also need to know what each macromolecules are made out of. Now, molecules are made out of atoms, right? We need to know atoms are forming the molecules. Now, carbs are made out of C, which is carbon. Easy, right? Carbs, so carbon. H, which is hydrogen. Easy, right? Carbon, hydrogen. And we have O, which is oxygen, okay? Now, CHO, I mean, it's not hard to remember, but it's going to be a bit more complicated when you learn because we have 3 more macromolecules to learn. And once you start learning all the atoms that compose each macromolecule, it's going to be complicated. So one easy way to remember CHO for carbs is just remember this. Carbs are in rice, right? Asians eat a lot of rice. And Cho is a very common Asian last name. I have a friend named Daniel Cho. I don't know. There are a lot of Chos, right? So just remember CHO. Cho is eats rice, and rice is carbs, right? So carbs, Cho, CHO. Second, lipid. Now, this time, let's start with all the atoms that compose lipid. Let's go opposite order. So lipids are created out of CHO, just like carbs, but you have one more, which is P. Okay, so CHO and P compose lipid. Now, how this? Now think it this way: lipid in colloquial language is fat. Okay, now fat is bad, right? We want to chop out all the fat from our body to be more defined, right? So lipid, fat, we chop it out. CHOP, chop. Okay. Now let's move on. What's a monomer? Monomer of a lipid. Well, it looks like this. And this part is called a glycerol. And this part is called fatty acid. Easy to remember, right? I mean, fat. Lipid is fat, so fatty acid. And what's a polymer of a lipid? Well, you don't have one. That is why you don't have a name for a monomer, but I just literally draw a structure, right? Because all lipid is a monomer. So there is not a specific name for a monomer, but it's just lipid. And there is no polymer for a lipid. Now, that does not mean that lipid does not bond to each other, okay? These monomer, these individual lipid pieces will still bond to each other to create a structure. And that leads to two different types of lipids. Based on the structure, you will have two different types of lipids. First is saturated. Second is unsaturated. Now, I mean, think about it. What does saturated means? We often say that job market is saturated. This liquid, this solution is saturated. That room was saturated with people. When we say saturated, it means full, right? It means packed. Saturated lipids are packed lipids. It's packed with all these monomers. They're tightly packed very closely and very tightly. Now, again, if something is packed, if molecules are packed, they can't really move, they can't really flow, meaning that it's solid. That is why saturated fats are solid fats. For example, butter. Okay. Now, unsaturated fat is it's not tightly packed. It's like it's kind of loose, like you have some empty area between each lipid. Now, if you have empty area, they can flow, meaning that it's a liquid. For example, olive oil. Olive oil can flow, it's liquid, right? So saturated fat and unsaturated fat. Now, what's really important to remember is why is saturated fat saturated? Why can it be packed in a small area, whereas unsaturated fat has those empty areas? Well, that is because saturated fats are made out of these fatty acids that only consist one type of bond, which is singular bond. Now, you don't have to know what singular bond, just know it's made out of one type of bonding. These fatty acids are made out of one bonding, and that is why it's so regular. Whereas when you look at these unsaturated fats, as you can see, I draw the fatty acids kind of weirdly, right? I kind of tweaked at the end. Why? Because in unsaturated fats, the fatty acid includes two different types of bonding, singular bonding and double bonding. Because it has different types of bondings, it looks kind of weird. Its structure is like weird like this. And because the fatty acid structure is weird, it can't be packed into a small area. Think about it. If you have the exact same looking Lego blocks, you can just pack them, you can just stack them, build them into one regular, very packed, very closely knitted structure. Whereas if you have all different sorts, all different shapes of Lego blocks, I don't know, like the weird shaped Lego blocks, you can't really fit them into a small area, right? That is why unsaturated fat cannot be packed into a small area to be a solid. And that is why it's always liquid. Okay, so just remember that. Now, there's one important characteristic that you have to know about lipids. Now, lipid fat is hydrophilic. Now, what does hydrophilic means? Well, hydrophilic means sorry. I'm so sorry. Hydrophobic. Hydrophobic. What does hydrophobic means? Well, think about it. Homophobic. Something phobic. Fat phobic. Always when you add, those were probably the worst examples I could have come up with. Sorry. But whenever we say something phobic, we're saying we're scared of something, right? Hydrophobic means you're scared of, mean that it does not like water. Fat does not like water. That is why on water, if you add some fat, if you add some olive oil, it's just gonna float on the top, right? Why? Because olive oil is unsaturated fat. Fat does not like water. Now, this does not mean that saturated likes water. Saturated fat would also hate water. Say you dissolve butter and you spray on, you dissolve butter and you just pour on water. It's still gonna float. It's not gonna mix because it wants to be separated. Okay. Now there are two more types of lipids that I want you guys to know. Of course, I mean we already know what fats are, right? You know the white part in beef or pork or whatever meat, or olive oil, butter, whatever. We know different types of fats, but there are two different types of lipids that you might not know as a common sense. First is cholesterol. Second is steroid. Okay. Now these two are also a type of lipid. So, and it might come up, so just know, okay? I mean, now that I'm thinking, cholesterol, steroid, butter, all the things that are bad for your body, it's always lipid. Lipid is the enemy. Now, does that mean lipid does not do any good to our body? Of course, no. If it has no benefit, then by natural selection, it would have just naturally, be not included from our body, right? Why do we still have fat? Well, I mean, think about it. Fats, first of all, it maintains your body temperature, homeostasis, very important. Also, fat is technically a form of storing energy. So when you're starving, your body will sort of break down fat and use that as an energy source. That is why when you want to go on a diet, when you want to lose weight, you don't eat, because when you don't eat, you're not getting energy source from outside of your body. So you have to break down energy from inside of your body, which makes your body break down the fat and use that as an energy source and make you look more jacked. Okay. Now let's move on to my very favorite type of macromolecule, which is, of course, protein. Let's go. Protein. So, again, what is protein made out of? It's made of carbon, hydrogen, oxygen, and sulfur. CHOS. Okay. Now, how are we going to remember this? Well, whenever you go to Subway, the server would ask you, so what protein would you like to choose? Choose your protein, right? Turkey, ham, chicken, beef. What protein are you gonna choose, right? You always choose your protein. So CHOOS, choose, and then talking about that. Okay, choose CHOS. CHOS, carbon, hydrogen, oxygen, sulfur, composed protein. Okay. Now again, what is the monomer? Monomer of a protein? Well, amino acid. Okay. Now there are 20 different types of amino acid based on their R group. Now, what is an R group? I'll explain later, okay? But just know that there are different types of monomers, okay? There are different types of amino acids. But what is a polymer of protein? Well, that is called a polypeptide chain of amino acids, okay? So, well, and polypeptide chain is made out of peptide bond. Okay, so polymers are made out of polypeptide chain, which is made out of peptide bond. Okay, now first of all, I'll explain how amino acids look like. Now, amino acid looks like this: amino group, carboxyl group, H, and then R group. Now, H is literally hydrogen. You just have one random hydrogen sticking out. Okay, and you have amino group, carboxyl group. Okay, now how does amino group look like? Normally amino group is on the left. It's always on the left. So just whatever is on the left, that's going to be amino. Okay, so don't worry. Whenever you, because in some questions they're going to give like weird chemical formula for amino acid, like CH blah 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 blah, in a very complicated form. But just remember, amino acid is always on the left. Okay, carboxyl group, it always contains C, it always contains carbon. So carboxyl, carbon, right? So whatever has a C, that part will be carboxyl group, but you don't have to remember that. Why? Because carboxyl group is always on the right. So whatever is in the right will be your carboxyl group, okay? R group is always on the the down part of amino acid. And this Amino group H and carboxyl group is all same for every single amino acid, but this R group is different. And this R group is the part that defines, that distinguishes each monomer or amino acid. So again, there are 20 different types of amino acid, which means there are 20 different types of R groups, okay? So just remember that. Now, when we're talking about polymer of a protein, it's very complicated because there are four different structures of a polymer or polymer of a protein. The first level, it's called primary. And this is literally just peptide bond. Of course, if you have an amino acid, it's going to be connected to another amino acid, right? And that's again going to be connected to another amino acid, and that will continue, right? This bond, this linkage between these amino acids is called a peptide bond, and that is the primary structure. Now, secondary structure of a protein polymer is called it's called a secondary structure, and it is formed by H bonding. Now, what happens is, as you can see, there's this one random H sticking out, right? This one random H forms hydrogen bonding between different amino acids. Again, hydrogen bonding is an intermolecular force. It's a force between two different molecules. So H possessed by one amino acid and H possessed in another amino acid will cause hydrogen bonding, and they will sort of pull each other, right? And this force of pulling, this force of attraction will make these polypeptide change fold. Why? I mean, if you have an H bond going on between these two amino acids, then they would want to stick to each other. They would always want to just, be around with each other. So they will pull each other. And so this polypeptide change will change its structure by folding. Now, third structure is called tertiary structure. And this is also a form of folding. But this time, it's not the H bonding that is folding the polypeptide change, but this time it is the R group that is folding the structure. How does that happen? Well, R group, some of them, out of 20 R groups, some of them are hydrophobic. How can it be hydrophobic? My friend likes water. Hydrophobic, okay? And some of them are hydrophilic, okay? So when you have R groups that are hydrophobic, then those R groups, when you have these peptide structure, those R groups want to sort of, be in the middle of the structure so that it doesn't have to meet water because they're scared the connection is not stable. Let's wait until the lighting is over. So look at this drawing, okay? Hydrophobic R group possessed amino acids would want to be folded towards more interior part of the structure because they don't want to touch water, whereas hydrophilic amino acids would want to stick on the exterior, on the outer layers. They can actually touch water because they love water, okay? So that is why R group, because of their hydrophobic or philic trait, causes this amino acid, this polypeptide structure to fold. Now, then you might ask me, so you have two different types of, to explain it this way. Secondary structures, secondary forms are folded on a micro level because of the wrinkles, right? But then when we actually fold your shirt to put in your closet, you have to fold on a more macro scale, right? So I would say that tertiary folding is more like the macro scale of folding the general shape of your shirt, whereas secondary bonding, secondary structure is more like the smaller wrinkles on your shirt that does not affect the bigger scale structure, shape of your shirt or your polypeptide chain, but it makes that smaller wrinkles here and there, okay? So that's why we distinguish the two. Last, we have quaternary structure, and quaternary structure is basically multiple amino acid chains. So through these three different structures and bondings and foldings and everything, now we have an amino acid chain made like this over here, right? But then we're not just having one amino acid chain, we're having multiple of them. And those amino acid chain will all, bond to each other as one structure. And now we finally have like this, all of them will connect to each other. And once all of them are connected, and by the way, all of them has their own wrinkles and macro foldings, will all connect to each other to create this bigger quaternary structure of a polypeptide chain. Okay, so I mean that's pretty much it with proteins. Well, no, sorry, I was lying. That is not pretty much it with proteins, because you have to remember two things. First is denaturation. Now, because protein is made out of these four very complicated structure, it can also be, the structure can also change. And that process of structure changing is called denaturation, okay? Now, denaturation happens when heat, pH, salinity. Now, salinity is salt, how salty or not salty the environment is. These things will change the structure. I mean, think about it. When you put a chunk of meat on your pan, heated pan, the colors got changed to brown from red, right? So you can see how it's changing when heat is added. Same with pH and salinity, right? So that is denaturation. Proteins change its structure when the environment changes. What's important is though, four of the protein structures are connected. They are all, reacting with each other. Meaning, if one small part in the primary structure changes, then the secondary structure will also change, which will cause tertiary structure to also change, and ultimately the quaternary, the big actual protein, will also change. And the biggest example is sickle cell disease. Sickle cell disease is when one amino acid from your primary structure, right? If you have these primary structures, one amino acid is wrong. It's a wrong amino acid. It's a very small mistake, but the small mistake will lead to an issue in folding and in macro folding, and ultimately in the bigger scale of how the protein looks like, which will make your body very ill. So that is sickle cell disease. Sickle cell disease is a very big, very typical example of how one small change in primary structure impacts the whole function and trait of the protein. Okay, so that's it. Now let's move on to the last part of this lesson. This video is nucleic acid. Now, nucleic acid is made out of CHO, carbon, hydrogen, oxygen, nitrogen, and P, which is phosphorus. So how are we going to remember? I was trying to think about a lot of different ways to make this easy to memorize. I really couldn't. I mean, just chomp. It sounds good, right? Nucleic acid is the longest one. It has the most diverse atoms composing it, but just remember it's chomp, okay? Just nucleic acid is chomp. What is nucleic acid's monomer? It is nucleotide, okay? Then what is its polymer? It is nucleic acid, literally just nucleic acid. Different types are RNA and DNA, only two of them, okay? Easy to remember. Now, how does a nucleotide look like? Nucleotide looks like this. You have a P group, phosphorus group. You have A, G, C, T, and then you have this oxy or deoxyribose, okay? Now, based on, if it is an oxyribose, it's going to be RNA. If it's a deoxyribose, it's going to be DNA. Deoxy, so it starts with D, so DNA, right? So it's R, so it's going to be RNA, okay? Now, how does then each monomer connect to another monomer to make a polymer? Well, that happens through H bonding, like this. Between this P group and this base, there is a hydrogen bonding, which connects the two of them. And that will continue going on like this, right? Now, this is RNA, right? You only have one strand, but DNA would have another strand, right? That looks like this. And we know that the two bases, AGCTs, will connect to each other. Then how do they connect? Is that also hydrogen bonding? Yes, this is hydrogen bonding. So hydrogen bonding connects AGCT to each other like this in DNA only, right? Not in RNA, because RNA is just singular strand, and when you have both strand, that is when it's a DNA, right? So one important thing is, again, I made a mistake. What connects each of them on one singular strand? I said it's a hydrogen bonding, right? That was a lie. It's actually a dehydration synthesis, okay? So what's interesting is you have both hydrogen bonding and dehydrogen bonding in DNA, right? So when you think about DNA, well, what I'm saying right now is that this base and this base are connected through H bonding, but this base and this monomer are connected through dehydration synthesis, okay? And how each base looks like is it looks like this, okay? So let's remember that. Now I'm going to explain the difference between DNA and RNA, okay? So DNA, RNA, by now you probably should know this, but DNA has deoxyribose, whereas RNA just has ribose base. DNA has AGTC, whereas RNA has AGUC. U is uracil, T is thymine. RNA's main purpose is copying. Well, only if it's a tRNA. It's mRNA, copying information, genetic information, whereas DNA's main purpose is storing genetic information. And lastly, DNA has, of course, two strands. RNA only has one strand. RNA is what it looks like this. DNA is what it looks like this. Of course, there are different types of RNAs, and I will explain that in later units. So don't worry about that. Now, that is pretty much it for all the macromolecules and water. Of course, I would advise to make a small chart, a table where you store all the information, right? What the monomer is, what the polymer is, what atoms make each of them, whether there's chop, chomp, right? Choose, cho, whatever. Also, how it is used in our human body and its structure. So that would be pretty much enough for you for this unit. And that's it. Thank you so much for watching. And most importantly, don't forget to have a great day.